Hello friends, today in this video I am going to discuss about the MCQs on general microbiology, and this is the 8th part of the series of MCQs on general microbiology. So let's start the question answer session for microbiology with explanation. So the first question is: mutation which occur in the absence of known mutagenic agent is known as missense mutation, spontaneous mutation, induced mutation or nonsense mutation. And the right one is option B, that is spontaneous mutation arise in the absence of any kind of mutagenic agent, and the spontaneous mutation arise occasionally in all cells and are often the result of error in DNA replication. Okay, and this is also known as random mutation, or it causes random changes. Okay, and this error in replication which cause point mutation other occurs can lead to frameship mutation. Okay, so move on to the next one, that is mutant strain in which phenotypic change involves the requirement of a growth factor is called wild type strain, phototropes or autotropes. And the right one is option C, that is auxotrope. So auxotrope mutant strain where phenotypic change involves the requirement of a growth factor. So auxotrope require a growth factor for their growth. So here we can see auxotroph are organisms that are unable to produce an essential organic compounds due to a gene mutation. So they have a mutation, that's why they cannot produce their own nutrient. Okay, and have an additional nutrient requirement. So they are mutant and have a mutation. And auxotroph cannot grow on minimal medium. So they require a certain supplement for their growth, and they are useful in figuring out metabolic pathways. As well as the prototropes are here. Prototropes means prototropes are organisms that are capable of producing all organic compounds needed for their growth. They are wild type. Okay, so move on to the next question, that is bacterial antibiotic resistance is frequently transferred by all of the following except transposon, R factor, intron, bacteriophage, and the right answer is option C, that is intron. So here we can see this is a bacterial DNA or any DNA. So this is not a bacterial DNA, it is a DNA in higher eukaryotes. So here we can see exons and introns are present. Intron is the non-coding gene and exon are the coding genes, because after transcription the DNA produce the RNA with exons and introns present in their DNA. But in next, after splicing, only the mRNA, only the exon parts will attached with each other and introns are exit out, and this only the exons will transcribe or code the DNA and produce the amino acid chain and then produce the protein. So introns are non-coding DNA. So that's why bacterial antibiotic resistance is frequently transferred by transposon, R factor, bacteriophage, but not by the introns. Okay, so next move on to the next question, that is genetic engineering uses all the following procedures except fine structure mapping, synthesis of DNA by DNA polymerase, cleavage of DNA by restriction endonuclease, or meiotic segregation. And the right answer is option D, that is meiotic segregation. And here we can see this is a chromosome, and in meiotic segregation at first the chromosomes are separated from each other, that is homologous chromosome separated from each other, and in next the sister chromatid segregate from each other. Okay, so this is the meiotic segregation. And the next one is bacteria which does not fulfill Koch's postulate is Corynebacterium, Gonococci, Mycobacterium or Salmonella, and the right answer is option B, that is Gonococci. Gonococci do not follow the Koch postulate. What is Koch postulate? Koch postulate are a series of ground rules. These are the rules to determine whether a given organism or bacteria can cause a given disease. So the Koch postulates are some parameters by which we can decide one organism is a pathogen or not for a particular disease. Okay, So move on to the next question, that is bacterial spore shows all the following structure except core, spore cortex, spore coat or capsule, and the right answer is option D, that is capsule. So bacterial spore do not contain the capsule. Okay, so here we can see the outer, so this is the bacterial spore structure. This is the outer part is the coat, then this is the outer membrane, and this is the cortex. Here this one is the cell wall, and this one is the inner membrane, and this is the core where DNA is present. Okay, so here no capsule is present in the bacterial spore. So next one is viable count of bacteria is done by pore plate method, lawn culture method, stroke culture method or streak culture method, and the right answer is option A, that is pore plate method. So viable count is done by pore plate method, and here we can see the pore plate method. So at first the bacterial sample is mixed with the agar, warm agar. The bacterial sample is mixed and then sample poured onto the sterile plate, and then the sample stirred to mix and allow to solidify. Okay, and then plate incubated until the bacterial colony grows. So this is the pore plate method. So move on to the next question, that is all are selective media for Salmonella species except deoxycholate citrate agar, xylose lysine deoxycholate agar, bile salt agar or Salmonella shigella agar, and the right answer is option C, that is bile salt agar. So bile salt agar is not a selective agar for Salmonella, because this thiosulfate citrate bile salt agar is a selective media for Vibrio cholerae, because the high pH which inhibit gram positive bacteria and most of enteric bacteria. Okay, so next one is CATGUT is sterilized by autoclaving, ethylene oxide, sodium hypochlorite or ionizing radiation. Right one is option D, that is ionizing radiation is used for sterilizing CATGUT. What is CATGUT? Is CATGUT, also known as gut, is a type of cord that is prepared from the natural fiber found in the walls of animal intestine. Okay, and CATGUT makes as usually use sheep or goat intestine. Okay, CATGUT makers usually use sheep or goat intestine. And the next one is, and here we can see ionizing radiation includes X-rays and gamma rays, cause mutation in DNA and produce peroxides in the bacterial cell. Okay, and these are commercially used for sterilization of disposable items, and this ionizing radiation is also called cold sterilization. Okay, and this ionizing radiation by gamma rays mostly done by the cobalt 60 radioactive element. So next one is flash method for pasteurization of milk is done at a temperature of 72 degrees Celsius for 20 minutes, 72 degrees Celsius for 20 seconds, 63 degrees Celsius for 30 minutes or 63 degrees Celsius for 20 minutes, and the right one is option B, that is 72 degrees Celsius for 20 seconds. And this flash method of pasteurization also called high temperature short time processing, because here high temperature is used with for short time, and it is a method of heat pasteurization of perishable beverages like fruit and vegetable juices, beer and dairy products. Okay, where we use 71.5 degrees Celsius to 74 degrees Celsius for about 15 to 30 seconds. Okay, so move on to the next question, that is sharp instrument are sterilized by autoclaving, 2% glutaraldehyde, 5% cresol or 70% alcohol, and the right answer is option C, that is 5% cresol is used for sterilizing sharp instruments. And the next one is SPHEROPLASTs are derived from gram negative bacteria, or they totally lack cell wall, or are produced artificially by lysozyme, or are not osmotically fragile, and the right answer is option A, spheroplasts are derived from gram negative bacteria. So the spheroplast and protoplast, which are a form of bacterial cell which don't have any, they are lack of cell wall. So the protoplast means the gram positive bacteria which lack cell wall is known as protoplast, and the spheroplast is gram negative bacteria which lack cell wall, that is called spheroplast. Okay, so move on to the next question, that is FIMBRAE can be detected by negative staining, modified acid fast staining, silver impregnation staining, or hemagglutination test. And the right answer is option D. So FIMBRAE can be detected by hemagglutination test. So these are the FIMBRAE. These are the FIMBRAE of bacterial surface, and they helps in addition to other cells and surfaces. And what is hemagglutination test? This hemagglutination test where hemagglutinin virus or hemagglutinin bacteria react with the red blood cell and agglutination occur, okay, or they agglutinate, or and in negative reaction, non-hemagglutinin virus or non-hemagglutinin bacteria cannot attach with the red blood cell or react with the red blood cell, so no agglutination done. Okay, so move on to the next question, that is oxidation fermentation test is done for indication of Shigella dysentery, Proteus mirabilis, Pseudomonas aeruginosa, Enterobacter erogens. And the right answer is option C. Pseudomonas aeruginosa is used for Pseudomonas aeruginosa oxidation fermentation test is done. So here we can see in a positive reaction the green color of the culture is transformed into a yellow color for the fermentation, where fermentation is done. So fermentation can be done in presence of oxygen or in absence of oxygen. So here this is a oxygen is present, but to make the culture or make the environment anaerobic, so we add the paraffin oil in this tube. So this is a non-anaerobic tube and this is a aerobic tube. Okay, so here we can see no fermentation done in here and here also. So this is a oxidative negative and fermentative negative. So here we can see this is oxidative positive and fermentative positive, because in presence of air and in absence of air, in both condition fermentation occurred. Okay, and here we can see this is oxidative positive plus but fermentative negative, because here in the presence of air oxidation done or fermentation done, but here in absence of air the fermentation cannot happen. Okay, so this is the process of oxidation fermentation test. And the next one is ZIEHL-Neelsen stain was discovered by ZIEHL-Neelsen, Paul Ehrlich, Joseph Lister, L. Mcqs. And the right answer is option B, that is Paul Ehrlich. And this ZIEHL-Neelsen test is also known as acid fast staining, okay, for stain the Mycobacterium tuberculosis. Okay, and this ZIEHL-Neelsen staining or acid fast staining, here is the procedure. So at first we apply the first or primary stain, that is the carbonyl fusion, for 30 seconds, and then heat fix the cell to the slide, and then we have to decolorize the decolorize with acid alcohol for 15 to 20 seconds, and then we add the counter stain, that is methylene blue. So the acid fast organism retain the stain of carbonyl fusion, that is the primary stain, and they appear as pink, and the non-acid fast organism appear as blue for their methylene blue counter stain. Okay, so move on to the next question, that is bacteriophage was discovered by Robert Koch, Louis Pasteur, Todt and Deherel, and Landsteiner and Popper. And the right answer is option C, that is Todt and Deherel discovered the bacteriophage. And next one is Louis Pasteur was a French chemist, French physician, German chemist, German physician. And the right answer is option A, French chemist. And here we can see his contribution to microbiology. So he was a French chemist, and he was known as the father or founder of modern microbiology, and he introduced fermentation, and he told that fermentation is a microbiological process. And he said beer and wine not produced without microbes, and he showed microbes cause fermentation and spoilage, and he introduced the pasteurization process or pasteurization technique, okay, and he disproved spontaneous generation of microorganism. He disproved the spontaneous generation theory of microorganism or living cell, and he developed aseptic techniques and developed a rabies vaccine. Okay, so these are the contribution of Louis Pasteur. And the next question is, 1 nanometer is 100th of the millimeter, 1000th of the millimeter, and 10,000th of the millimeter, or 1 millionth of the millimeter. And the right answer is option D, 1 millionth of the millimeter. So 1 million means 10 to the power 6 and 1 millimeter means 10 to the power 3. So if we add it, so we can see that 1 nanometer is 10 to the power minus 9 meter, 10 to the power 6 and 10 to the power minus 3, so 10 to the power minus 9, so 10 to the power minus 9 meter. 1 nanometer is 10 to the minus 9 meter. Okay, so move on to the next. That is, all are fluorescent dyes used in fluorescence microscopy except leishmine, rhodamine, Congo red, or fluorescent isothiocyanate. And the right one is option C, that is Congo red is not a fluorescent dye. And this Congo red is used for staining amyloids and for the cell wall of plant and fungi and for the outer membrane of gram-negative bacteria. So move on to the next. That is, all the following are acid-fast organism except Mycobacterium leprae, nocardial species, bacterial flagella, or bacterial spore. And the right answer is option C, bacterial flagella. So bacterial flagella is not an acid-fast organism. So what is acid-fast? Acid-fast organisms have a lipid capsule that has a high molecular weight and is waxy at room temperature. So this makes the organism impenetrable by aqueous based staining solution. So the lipid capsule of a acid-fast organism stain with carbonyl fusion and resist decolorization with the dilute acid rinse. Okay, so move on to the next question. That is, flagella of Vibrio cholerae is monotrichous, lophotrichous, amphitrichous, or peritrichous. So flagella of Vibrio cholerae is a option A, that is monotrichous, and monotrichous means only one flagella is present in one end, and amphitrichous means in both sides one flagella is present, and lophotrichous means in one side multiple flagella is present, and in peritrichous in whole body or whole periphery flagella are present. Okay, so next question is, pili of bacteria helps in locomotion, adhesion, protection in unfavorable condition, or protection against antibacterial substances. And the right answer is option B, pili helps bacteria for adhesion. Okay, here we can see these are the pili. Through pili we can see the conjugation happened by this pili also. So move on to the next question. That is, Campylobacter jejuni is obligate aerobe, facultative anaerobe, obligate anaerobe, or microaerophilic. And the right answer is option D, microaerophilic. So Campylobacter jejuni, which is present in our stomach or intestine, they produce they are microaerophilic nature. Okay, so move on to the next question. That is, so here we can see a microaerophilic is a microorganism that require environments containing lower level of dioxygen that are present in the atmosphere for optimal growth. So in basically in environment there are 20% of oxygen are present, but they require only 2 to 10% of oxygen for their growth. So that's why they are microaerophilic. So here we can see that bacteria are differentiate them into aerobic bacteria and anaerobic bacteria, and this aerobic bacteria divided into obligate aerobe who cannot survive without the oxygen, and the microaerophile which require who who require lower concentration of oxygen like 2 to 10%, and obligate anaerobes means they cannot survive in the presence of oxygen, and facultative anaerobe means this bacteria can grow without oxygen but use oxygen if it is present there. Okay, and aerotolerant bacteria means this bacteria cannot use oxygen for their growth but are not harmed by it. That means they are tolerant to air. Okay, resistant to air. So move on to the next question. That is, Loeffler's serum slope is sterilized by sterilized in hot air oven or in autoclave or by inspection or by tinderlization, and the right answer is option C, that is by inspection. And this inspection is a fractional sterilization. That means this process of heating an article, this is the process of heating an article on 3 successive days at 80 to 85 degrees Celsius for 30 minutes by an inspirator. Okay, and the method kills all vegetative forms of spores. It is used to sterilize egg-based media such as Lowenstein, Janssen or LG medium, and Dorset's egg medium, and serum-based media such as Loeffler's serum slow. Okay, so the last question is, in gram staining, spores appear as blue colored bodies, purple colored bodies, red colored bodies, or refractile bodies, and the right answer is option D, that is refractile bodies. So in gram staining, spores appear as refractile bodies because they are called refractile bodies because they are greater density than the rest of the microorganism's body mass and cause light to be refracted or bent when it is passed through them. Okay, so here we can see this is the gram staining, and here this is the gram staining of bacteria, and here this is the spore. This white color is the spore. Okay, so these are the all questions for general microbiology. Okay, thank you for watching this video.